Group research attention deficit hyperactivity disorder, also known as ADHD. The CDC defines ADHD as one of the most common neurodevelopmental disorders of childhood. Children with ADHD may have trouble paying attention, controlling impulsive behaviors such as acting without thinking about the possible consequences, or being overly active. ADHD occurs in about 5% of children with little geographic or cross-cultural variation in prevalence.
In relation to inheritance, males are almost 3 times more likely to be diagnosed with ADHD than females are. Children living in households where English is the main language are more than 4 times as likely to be diagnosed as children living in households where English is the second language. ADHD also has a tendency to run in families, but the inheritance pattern is usually unknown. Overall, the risk of developing this condition is about 9 times greater for first-degree relatives.
People with ADHD are at risk for many functional impairments such as school failure, peer rejection, injuries due to accidents, criminal behavior, occupational failure, divorce, suicide, and premature death. Neuropsychological studies implicate brain circuits regulating executive functioning, reward processing, timing, and temporal information processing. ADHD often co occurs with other conditions, including mood, anxiety, conduct, learning, and substance use disorders. Longitudinal studies also show that 2/3 of ADHD youth will continue to have impairing symptoms of ADHD in adulthood.
There are many effects that come with ADHD. For example, children with ADHD appear to have nearly 50% lower levels of an amino acid called tryptophan. Tryptophan helps in the production of dopamine, noradrenaline, and serotonin. The DBH gene provides instructions for producing the enzyme dopamine beta hydroxylase. This enzyme converts dopamine to noradrenaline, both of which are chemical messengers, aka neurotransmitters that transmit signals between nerve cells. Noradrenaline plays an important role in the autonomic nervous system, which controls involuntary body processes such as the regulation of blood pressure and body temperature.
One of the main altered proteins involved with ADHD is the BDNF gene, which provides instructions for making a protein found in the brain and spinal cord called a brain-derived neurotrophic factor. This protein promotes the survival of nerve cells by playing a role in the growth, maturation, and maintenance of these cells, and is also very important for learning and memory. The BDNF protein is found in regions of the brain that control eating, drinking, and body weight. The protein likely contributes to the management of these functions.
Our genetic test is testing only a few different genetic variations, specifically the SNMP versions of RS8079626 and RS7210438. There are GC haplotypes which have a slightly lower risk and AC haplotypes that have a slightly higher risk for adult ADHD. The two RSGC haplotypes affect the BAIAP2 gene on chromosome number 17 and encodes for the insulin receptor tyrosine kinase substrate protein of 53kDa, which creates a lack of excitatory synaptic signaling, which then displays a lack of social interaction and communication, which would lead to the indication of the gene for ADHD being present.
Some general overall benefits of ADHD genetic tests would be that clinical intervention may improve outcome, family members at risk can be identified, positive health behavior can be reinforced, and there's an overall reduction of uncertainty. Our test results can provide a sense of relief from uncertainty and help people make informed decisions about managing their health care. A negative result can help eliminate the need for unnecessary checkups and screening tests in some cases. A positive result can help direct a person towards available prevention, monitoring, and treatment options.
Some general overall limitations or risks of our ADHD genetic test would be a possible adverse psychological reaction, cause of family issues or distress, uncertainty due to incomplete penetrance, insurance or job discrimination, confidentiality issues, and intervention also carries risk. One main limitation of our SMP genetic test for ADHD is that there are only 3 SMP variations for the gene. One gene, BAIAP2, has been significantly proven to have a connection with ADHD. The formal heritability of ADHD is about 80%, making it higher than any other psychiatric diseases, even though there are way fewer SMP variations at only 22%.
One implication of testing positive for a genetic disease would be its overall effect on your emotional or mental health. It would increase your stress and anxiety as well as your loved ones due to uncertainty or inconclusive test results. There is nothing you can do to prevent yourself from being positive, so taking a genetic test does not eliminate a person's risk for the disease. Another concern is the financial aspect. Genetic testing can be very costly and unaffordable. The test could also possibly reveal an inherited mutation that puts themselves or their kids at risk, since each parent contributes 50% to a child's DNA, which could lead to guilt and feeling responsible for your children's suffering. Genetic testing predicts the risk of disease or illness, which is serious information that is often misused. Employers and insurance companies have been known to deny individuals essential health care or employment based on knowledge of genetic disposition. This type of discrimination can be socially debilitating and have severe socioeconomic consequences. This limitation could negatively affect someone physically, mentally, and financially.
These are the citations for the sources we used and referenced in our assignment. These are the acknowledgements of each group member's contribution to the project overall.